the actual presentation. So how should we, first thing is that what is a pigment and how should we define the pigment? Okay, so in chemistry, pigment is any organic or inorganic molecule or compound that imparts the color to a particular material, and it is insoluble in chemistry definition. And in biology, the pigment is any substance that offers color to an organism or to a particular material, but difference is that here solubility, despite of its solubility characteristic. Okay, so the key difference is between their solubility. In chemistry, pigments are strictly insoluble in the medium, means that they do not dissolve but remain as fine particles suspended within the substance or material they color. Okay, for example, one example is there, like for example, titanium oxide. It is a white pigment used in paints, sunscreen, and plastic. Okay, so it remains suspended in the medium rather than dissolving in it. Okay, and in biology, pigments can be either soluble or insoluble. Like for example, soluble pigment, chlorophyll. Okay, it gets dissolved in the plant material. Okay, and one more example is melanin. It is insoluble pigment. So this is about the definition of pigment. Okay, now let's talk about the sources of pigment. Now pigments are generally obtained from living sources and non-living sources. Okay, so as you can see on the screen, sources of pigment, living sources and non-living sources. Okay, so in living sources of pigments, there are three classes. Okay, first is microbes, second plants, and third is animal. Okay, we'll start with the first class, which is a microbe. Okay, so in microbes, many bacteria, fungi, and algae, they are well known to produce the pigments. Like we'll start with the bacteria. Like you can see on the screen, the best example or well-known example of a pigment producing bacteria is a Serratia marcescens. Okay, it produces a red pigment which is called as a prodigiosin. The next example of bacteria which can produce a pigment is well-known example, I'll say, is a Pseudomonas aeruginosa. Now it produces a blue-green pigment which is called as a pyocyanin. And next example is many Streptomyces species, they can produce a blue pigment. As you can see on the right corner of the screen, it is actinorhodin. Okay. Now let's talk about this second category under the microbial class. Okay, it is a fungi. Now many fungi, they can produce the pigments. Specifically, the well-known example and very popular example is Monascus purpureus. Okay, now this fungi, it can produce a range of pigment which are called as a monascus pigments. Okay, which are red, orange, and yellow in color, as you can see on the screen. Okay, the second example is Penicillium species. Okay, now these Penicillium species, they produce many pigments which are green, yellow, and red in color. You can see this on the screen at the bottom of the screen. Okay, now next time example of fungi which produces the pigment is Aspergillus species. Okay, so this Aspergillus species, they produce, we all know, it produces a black and brown color pigment which is called as melanin. Okay, now let's talk about the third category under microbes, which is algae. Okay, now many algae, we know, they produce a diverse range of pigment. Best example, some well-known example is spirulina. Okay, it produces this algae, it produces a blue pigment which is called as a phycocyanin. The next example is Dunaliella salina. This algae, it produces a orange color pigment which is called as a beta carotene. Okay, and the next example in algae is Hematococcus fluvalis. Now this algae, it produces a red pigment which is called as astaxanthin. Okay, so this is about the microbes which can produce the pigment, well-known examples of the microbes which produces the pigment. Okay, now let's talk about the second class under the living sources of pigment, which is the plant. Okay, now we all know that plants, they produce pigment. Okay, best example is the chlorophyll, which is a green color pigment which is found in all green plants. Okay, next example is carotenoids, which are yellow, orange, red pigments. Okay, and we know these carotenoids, they are present in carrots, tomatoes, and peppers. Okay. Now, next example of plant pigment is anthocyanin, well-known example. Okay, which are red, purple, and blue in color, and they are generally found in berries, grapes, and flowers. You can see it on the screen at the bottom. Okay. Now, next example of plant pigment is lines. Okay, which are red and yellow in color, and which are generally found in beets and cacti. Okay. So this was about the plant pigments. Now let's talk about the animals pigments in short. Okay. So many animals, they can produce the pigments. Like best example, we all know melanin. Okay, which is present in skin, hair, and eyes of animals. Okay, you can see it in the screen, which is there on which is uh, which is there. Okay. So this melanin, it protects the animals against UV radiation. So this is the best example or well-known example of animal pigment. Now some more examples are there, like carotenoids. Carotenoids are also produced by some animals, like you can say sea fishes are there, shrimps are there. Okay, and that is why you have observed that some flamingos, they turn pink due to overeating or carotenoid rich eating food. Okay, so you can see on the screen that some shrimps, they can produce the carotenoid. Okay. Now the third example is hemoglobin and hemocyanin. Okay. Now we know that it is a blood pigment which is found in vertebrates. Okay. Some more examples are there, like sepia is there, which is also called as a ink pigment, which is secreted by cuttlefish for the defense. And one more example is there, which is secreted by cilantrazine. Okay, now this is the bioluminescent pigment found in the jellyfish. Okay, now let's talk about in short about the non-living sources of the pigment. So pigments, we can obtain pigment by many non-living sources like minerals and rocks. Example, the one red pigment is there, which is called as a hematite. Okay, and the pigment, green pigment, which we use in our laboratory, malachite green, and lapis lazuli, which is blue in color, they are generally extracted from the minerals and rocks. You can see the pictures on the screen. Actually, blue colored and green colored rocks, they are there. Okay, so we extract some pigments from the minerals and rocks, and so some pigments we synthesize artificially in the labs, like azo dyes are there, many other pigments are there. Okay, so this is all about the sources of the pigments. Now let's talk about the microbial pigments in detail. Why we prefer, why everyone talks about microbial pigments, and why everyone wants to work on microbial pigment. We'll talk about this. Okay. So actually, microbial pigments, they offer many advantages and benefits over other pigments. Okay, other pigments extracted from or obtained from plants, animals, or synthetic chemicals. Okay, like we'll discuss those benefits first of all. Benefits first of all, like first benefit is that these microbial pigments, they are eco-friendly and sustainable. Okay, now why do we call them eco-friendly and sustainable? Because first reason is that they are renewable source of pigment production. Okay, so as we know, microorganism can be cultivated rapidly and in large quantities using fermentation, which makes microbial pigments a sustainable alternative to plant or animal derived pigment. Okay, so this is the first reason. Now second reason is due to their low environmental impact. Okay, now microbial production of pigment, it generates, you can say, less waste and pollution compared to synthetic pigment manufacturing processes. Okay, so this is the second reason why we are calling them eco-friendly and sustainable. And the third reason is that we can use waste substrate for microbial production. So many microbes, we can grow them on agricultural waste or industrial waste, okay, which reduces the need for, you can say, using other raw material. Okay, so this is the last reason why we are calling them eco-friendly and sustainable. Okay, now next benefit or advantage is that these microbial pigments, they are non-toxic and safe. Okay, now Now, again, same thing. Why we are calling them non-toxic and same? Then first reason is that they are biodegradable. Okay, we all know, we all microbiologists, we know that microbial pigments, many microbial pigments, they are as they are naturally derived. Okay, and many break down easily in the environment, unlike other pigments like synthetic pigments that may persist and cause pollution. Okay, so the first reason is biodegradation. This second is non-carcinogenic. Mostly microbial pigments, they are non-carcinogenic, unlike synthetic dyes, like azo dyes and other dyes. They are carcinogenic and they are very harmful to the environment and all the living beings. So generally, these microbial pigments are non-carcinogenic, and that is why we are calling them non-toxic and safe for the living beings and for the environments. Okay, and the third reason is you can say they are suitable for sensitive applications, like we can use them in food industries, we can use them in cosmetic industries, and we can also use them in pharmaceutical industries due to their safety profiles. Okay, so that is why we are calling them non-toxic and safe, and this is the second advantage and benefit of microbial pigments over other pigments. Okay, now, next benefit and advantage is that microbial pigment production process, it is a cost effective. You can say it as a cost effective process. Now, why? Because first reason is that we can get high yield. Okay, so some microbes, not all, but some microbes can produce pigments in large quantities, but through controlled fermentation process. This is important. We should use this this controlled fermentation process, then only we can get high yield. Okay, now next reason why this process, microbial pigment production, is cost effective process is that a low cost substrate. Okay, we can use low cost substrate, as I have told you that we can use many waste materials like agricultural waste we can use for the production of microbial pigment. So this process, it is a low cost substrate process. Okay, and third is scalability. Okay, so scalability, we know that the fermentation processes, specifically microbial fermentation processes, are easily scalable for industrial production. Okay, so these are the reasons why we are calling the microbial pigment production as a cost-effective process. Okay, now next reason or next benefit of this microbial pigment production is that they possess additional functional properties, like many microbes, they possess antioxidant activity. Then some microbes, they also possess antimicrobial properties, protection properties. Okay, so this is the fourth benefit. Now, on screen, you can see the advantages or benefits of microbial pigments over synthetic dyes, plant pigments, and animal pigments. Okay, so if we'll talk about the synthetic dyes, as I have told you many things, but specifically in comparison with the synthetic dyes,
गलत लेके गई मैं
they are non-toxic, biodegradable, eco-friendly, and safer for human use. When we'll talk about the pigments, we'll compare this microbial pigment with the plant pigments. The main benefit is that faster production and higher yield. Okay, as we know, plant pigments, they are season dependent. Okay, so we get faster production, and they are we can say they are independent on season. Okay, and when we compare these microbial pigments with the animal pigments, so these microbial pigments, we can say they are ethical and sustainable. Okay, so this is all about the benefits of the microbial pigments. Now, let's talk about the characterization of microbial pigments. How should we characterize the microbial pigment? Okay, and which things are important to characterize the microbial pigment? So remember, characterization of microbial pigments, it involves physical properties, chemical properties, and biological properties. Okay, so what comes under the physical property of microbial pigment? So things like color. Okay, the shade of the pigment, okay, which is visible, like whether that pigment is red in color, yellow in color, blue in color. Okay, it comes under the color, physical property. Then second characteristic which comes under physical property is solubility. Okay, does that pigment dissolve in water or something else? Okay, it is solubility characteristic. Or for example, some pigment, they specifically dissolve they get dissolved in ethanol, methanol only. Okay, they do not dissolve in water. So all these things they come under the solubility characteristics. Third thing is a melting point. Okay, third the factor is a melting point. So we know that every molecule, every substance has a particular melting point. Okay, what is melting point? We know melting point is the temperature at which the pigment shifts from solid to liquid. Okay, so this melting point is under the physical properties. Then next factor is crystallinity, whether the pigment form crystalline or amorphous structure. Okay, and next two things are absorption and thermal stability. What is the stability under the wide range of temperatures? We should know. This comes under the physical properties. Now, what comes under the chemical properties? So if you want to characterize a given pigment based on their chemical properties, you should know about the molecular structure. Does whether that pigment it possesses polyketides, carotenoids, quinones, or what kind of rings it possesses. Okay, second thing, what kind of functional groups that pigment possess. Okay, does that pigment possess a hydroxyl group, a ketone group, or carboxyl group. Okay, so all these things you should determine in chemical properties. Then under chemical properties, mm, you should get to know about pH sensitivity, then oxidation reduction potential. Okay, how stable is your pigment under oxidative environments, and how it interacts with metal ions and acid is all. It also comes under the chemical properties. Okay, so this is all about the chemical properties of a given pigment. Now, next properties to characterize a given pigment, it is a biological property. So I have told you. So biological properties involves a study of antimicrobial activity, study of antioxidant properties. Okay, anti-inflammatory effects are there. Then some pigment they may possess cytotoxicity and anti-cancer potential. Okay, and some pigment they even they have been reported to have enzyme inhibition. So we should know whether a given pigment it has a potential to block a specific enzymatic pathway or not. Okay, so all these things we should study under the biological properties of a given pigment. Okay, now third factor which is very important, I will say which is very important in pigment research is pigment extraction. Okay, so you should know about two things. Okay, two kinds of solvents we require to extract a particular pigment. Okay, and to extract a particular pigment you should know the nature of your pigment. Okay, for example, if your pigment is hydrophilic, water soluble, it is if the nature is hydrophilic, then you should use polar solvents like methanol is there, ethanol is there, acetone is there, water is there. Okay, so you should go with the polar solvents. And if your pigment it is a hydrophobic, okay, then you should not select polar solvents. You should select non-polar solvents like chloroform is there, hexane is there. Okay, many other solvents they are also there. But you should know these two things that uh, if your pigment is hydrophilic, go with the polar solvent. If your pigment is hydrophobic, select the non-polar solvents. Okay, for the extractions. Now, this is about the extraction process. Now, let's talk about the important techniques in characterization of pigment. So, which techniques or what kind of techniques you need to characterize a particular pigment in your research? Okay, so first, an important technique is spectroscopic technique. And in that spectroscopic technique, first specific technique is UV visible spectroscopy. Okay, so we use this UV visible spectroscopy in microbial pigment characterization. Okay, so what it does, it measures the absorbance of light by pigment at different wavelengths, and so we can get the absorbance maxima. We know that absorbance maxima it is also called as a lambda max, and each pigment has a particular unique lambda max. For example, carotenoids they have a lambda max between the 400 and 500 nanometer. Okay, azaphilones, okay, for example, monascus pigments, they have yellow pigments, they have the lambda max value between the 330 to 450 nanometer. Okay, if the orange pigments are there, then lambda max value will be between the 460 to 480 nanometer. Okay, So likewise, we can determine the lambda max value of the given pigment. Okay, so next question is that why to perform and what knowledge do we get after using this technique? Why should we go with that UV visible spectroscopy? So first thing is that uh, you can identify or we can identify the pigment. Okay, how? So remember, each pigment has a particular lambda max. Okay, and by you can I can say, so what you can do, you can absorb, you can record the absorbance maxima, which is called as the lambda max of a given pigment. Then you can compare your obtained lambda max with the known lambda max available in the literature. Okay, and so this is how we can infer the type of pigment. Okay, so the first benefit is that you can identify the pigment. The second advantage of using this technique is that you can check the purity status and you can estimate the concentration of a given pigment. Okay, concentration, how you can estimate the concentration? By using, this is the basic in microbiology and other biology, using a Beer Lambert's law. Okay, we can determine the pigment concentration. And how can you determine whether the given pigment is pure or impure? So if you are getting multiple peaks or broad spectra, it means your pigment is impure, or you can say it is a mixture of pigment. Okay, so this is how you can get to know about the purity and concentration of the pigment. Next benefit of using this spectroscopy is that you can come to know about stability factors, like when we measure a pigment's UV visible spectrum at different pH and temperature, we can see how stable it is and whether its structure changes. Okay, so this is how its structure changes. You can say, or we can say that something is different. Okay, so whether that pigment is stable or not. Okay, so this is all about the UV visible spectroscopy. Now, second technique under the spectroscopy is FTIR. Now, FTIR, we know it stands for the Fourier transform infrared spectroscopy. So, why is it important? So, it is important because it is helpful in getting information regarding uh, structure of that particular pigment. Okay, so it helps in recognizing functional groups in pigment by measuring their infrared absorption at different wavelengths. Okay, so this is why the FTIR, it is very important in pigment characterization. Okay, so we can identify the functional groups which are present in the pigment. Like if you're getting a broad peak around 3200 to 3600, okay, you can say that hydroxyl, this group, it may present in that pigment. If you're getting a sharp peak between 1650 to 1750, okay, then you can say that pigment, it may contain or it is containing the carbonyl group. Okay, so depending on those peaks and by studying that spectra, you can say what kind of functional groups they are present in a given pigment. Okay, so what we know after this analysis, after using this technique, is that you can identify the functional groups, how I have told you that depending on the peaks, you can tell about the functional group. Second, you can confirm about its purity status. Okay, if you observe some kind of you can say unexpected peaks, if you observe in your spectra, then you can say that some chemical impurities or some byproducts, they are there in your pigment. Okay, and main thing is that, again, same thing, you can compare your spectra, your obtained spectra, with the standard spectra. Okay, so this is how you can compare or you can confirm the purity. Okay, again, same thing, third benefit, you can assess the stability. If you find something different spectra or something different peaks, okay, you can say that the given pigment, it is unstable under the given whatever maybe your temperature or pH you are studying. Okay, you can tell that it is unstable. Okay, so this is all about the FTIR. Now, the third technique which is useful in mass spectrometry, sorry, in the characterization, it is a mass spectrometry. Now, this mass spectrometry, it is helpful in determining molecular exact molecular mass or molecular weight and molecular formula of pigment. Okay, by measuring the mass to charge ratio of ionized molecule. Okay, so we can determine by using this mass spectrometry, we can determine the exact molecular mass and molecular formula. Okay, and the second benefit of this mass spectrometry is that it detects the fragmentation pattern for structural features. Okay, so we get it's same thing happens here. We get a spectra. Okay, specific ion, we get specific ion peaks, and these ion peaks, they can reveal us what kind of molecules or what kind of elements and what is the pattern of this element. All these things we get to know after doing this mass spectrometric analysis. Okay, the next and fourth important uh, technique for characterization is nuclear magnetic resonance, NMR, which is called as NMR. Okay, now this NMR, it is performed to determine, you can say, precise molecular structure of molecular pigment. Okay, by analyzing their atomic environment. Again, we can identify the functional group and their connectivity. This is important, functional group with and their connectivity. Then by using this NMR, we can differentiate structurally similar pigments. Okay, and third, we can understand the molecular dynamics and interactions of a given pigment. Okay, so what knowledge is gained after doing this analysis is that, see, there are two types of NMR, H1 NMR and C13 NMR. So this H1 NMR, it provides information on hydrogen atoms. Okay, so proton environments, we get to know about the proton environments, and C13 NMR, it identifies carbon skeleton and backbone. Okay, so this is all about the NMR. So these techniques, I'll say these techniques, they are very important in characterization of pigment. If someone is working, or some person or some researcher, they want to work on the microbial pigment, they should know about all these things like NMR, FTIR, UV visible spectra, lambda max, etc. Everything you should know. Okay. Now, last thing which I want to share is challenges in microbial pigment production. Trust me, working on microbial pigment production, it is not an easy task, because many factors, many things, they affect the microbial pigment production. I have worked on microbial pigment for near about for 5 years, and I have observed that temperature, pH, all these things, environmental factors, they affect a lot. Okay, many times we get stressed. What is happening and how is it happening? Okay, I have everything, but I now right now I'm not getting anything, not a single pigment you get. Okay, so these things, pigment production or pigment production process, it get changed depending on the factors, environmental factor. Okay, so challenges, first challenge, I'll say environmental factor, according to me, because pH, temperature, and nutrient conditions affect pigment production. So I would say there is a need to control all these factors strictly. We should monitor and control all these factors like pH, temperature, nutrient conditions strictly if we want to produce a given pigment with high yield. Okay. Now, second challenge is that some microbial strains, they may lose pigment producing ability over time and require genetic modifications for consistency. And really, trust me, this is also a common problem. Who are working in microbial pigments, they know that some strains lose their ability or their the capacity to produce a pigment. Okay, uh, reasons are different. Like reason, it may be due to genetic modification, mutation. Okay, but this is a challenge. Okay, the third is low yield and productivity. So many microbes produce pigment in very small amounts, so we need to optimize its production for large scale industrial applications. Okay, we can say. So low yield, another challenge. And last is scalability issues, like scaling up from lab to industrial levels without affecting yield, purity, or quality is challenging. So According to me, these four challenges, they are very critical if you are working or if you want to work on microbial pigment. So that's all from my side, sir. Over to you, ma'am.
Okay. Hello.
Yes,
If
ma'am.
somehow any doubt, please ask her. Now the session is open for the discussing. Hello. If someone has any query, please ask.
Very good presentation by Dr. Najia.
Thank you, sir. Thank you very much.
Compliments for a nice presentation and a very good insight with reference to the microbial pigment. You can hear me?
Yes, yes, sir. Thank you, sir. Thank you.
Oh, wonderful. Okay, what I actually came across with the two, three major issues. One is as it is a microbial product, the cost of production, media.
Yes, sir.
Another is the light sensitivity of the microbial pigment or any other
Correct.
pigment also,
Correct, sir. Correct, sir.
you know. So with reference to these two drawbacks, what is your opinion? The third thing I have just one observation with reference to the rhodotolar gracilis, which produces suppose one type of pigment, but upon mutation, one can have a different range of the pigment. So how it actually happens?
Yes, sir. First of all, I'll talk about those two factors like light affects the pigment production. 100%, sir. I've told you that all environmental factors, they affect the pigment production. I just said the temperature and pH, but light also affect the pigment production. And second thing was actually what you said was it was about the mutation. Okay, so
Yeah.
I have told you that genetic modifications, they may be you can say a reason for these kind of a change. Okay, so this is
Mhm. Yeah, but in that case, then we will not be much reliable on the microbial colors, right? If we have to have a scale up production or industrial scale up. So what is actually the strategy one should have that
that
you
is
get
why.
consistent product?
Yes, sir. That is
I
why
hope
I
you
told you that this is a someone is working and someone wants to work on microbial pigments. Please work on this thing. Like why that strain it get changed? Okay, what happens with that strain? If we get a consistent
Mhm.
strain or stable strain, then we can 100% we can do something great in microbial pigment production, sir.
Wonderful, wonderful. Thank you.
Uh, may I ask, please?
Yes, ma'am. Yes, ma'am.
Hmm. It was a very good presentation, and I think many people they are interested to work in the field of pigments. There is one of my friends who is sitting here. She's doing
Yes,
BTech, and she just asked me 2 days back that if I work on some project on pigments. So I just sent her your link, and she's there. I think in the group. Arlene,
Yes,
are you
ma'am.
there?
Yes,
Yeah.
ma'am.
So you can, I think you have a good chance to ask
Right,
from Nadia, ma'am, that whatever is your interest, and you can, share your feelings, right?
right.
And one thing, Nadia, I would like to know that
yes, ma'am.
these pigments you have been extracting from the cell surface or from the agar or from the broth.
Okay, ma'am. So the question is that how from where we should extract the pigment. So,
Yeah.
ma'am, actually, it depends on the nature of your pigment or what kind of pigment your organism is producing. If your organism it is producing the extracellular pigment, then you should extract it from the broth, okay, broth or whatever material is there, okay. And if your pigment, your organism, it is producing intracellular pigment, then 100% you should extract it from the cells only, okay.
So it must be well established by now that which organism is producing, intracellular or excreting the broth.
Yes, yes, ma'am.
You have some
Literature,
literature about that.
yes, yes, literature is available. Many have worked,
Because
and
Pseudomonas aeruginosa में तो आप ब्रॉथ में भी करते हैं तो उसमें भी कलर हो जाता है सारा
yes,
ब्रॉथ
ma'am.
भी.
Pseudomonas aeruginosa it
Hmm.
produces an extracellular pigment. Okay,
Yeah, yeah.
okay? So if you are working on Pseudomonas aeruginosa, then you should extract it from the cloth only.
Yeah, yeah. Okay. Thank you, Harleen. If you have any questions or feelings, please.
Ma'am, I wanted to ask, is there any industry currently that is using microbial pigments for production of maybe cosmetics or something, food industry?
Actually, research of microbial pigment production, okay, industrial use of these pigments, it is under research nowadays. So
Okay.
in near future, will uh, uh, come to know that yes, microbial pigments are also being used in industries.
Right,
Hmm.
ma'am. Right. Thank you so much.
So
Good evening,
Harleen,
ma'am.
you have good scope to work in this field. You had that hesitation that day that whether I should go ahead with this
Yes, yes.
project or
Actually,
not.
I was very double-minded because
Yeah.
I've planned to work on Pseudomonas, but Pseudomonas strain PW 20. So I didn't find much literature on it yet. Like there's some research done, but not to the end point. Like no one is using those pigments as such right now.
So Dr. Nazia might have now a lot of literature on this because she has completed her PhD in this field.
Right,
So I
right.
think she
Yes,
can
ma'am.
share
There is.
with you different types of literatures, and you can work together,
Yes,
isn't
Right,
you
it?
can
ma'am.
contact me. Yes, who is there? I don't know, but you can
Okay.
contact me. I'll provide
Yeah.
you material and I'll provide you literature.
Sure, ma'am. I'll get in touch with you through Dr. Nina.
Yes, yes. No
Thank you,
problem.
ma'am.
Thank you.
Yeah.
Good,
Okay.
good evening, Dr. Nazia. I am Dr. Purnima from Delhi University. And
Okay,
in
good
fact,
evening.
we are doing one undergraduate small scale project in our department. And some of my students are also working on this project are listening to you. So they must have got some insight into how to work. We have also got isolated a penicillium, which is giving a very beautiful red color. And
Yes,
we are in process of physical and chemical characterization.
yes.
So your talk has given us more insight that how should we go ahead in this. So thank you.
Thank you, ma'am. Thank you very much.
Hello,
Yes, sir.
hello, Dr. Najia. This is Dr. Sunil. I just
Yes.
want to know, is it true that every microbial pigment is non-toxic and safe?
No, 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 not all, sir. But mostly many microbial pigments, they are non-toxic. Not all microbial pigments, they are not toxic. Okay. Many are known to produce some kinds of toxins. So it is not correct, sir.
So for the primary study or for primary screening, how we can rule out that the isolated pigment
You
is
should
non-toxic?
check its pathogenicity, or you can check if whether your organism or your pigment it is toxic or non-toxic. You can study on that aspect, sir.
No,
Many
but
methods are there, many processes are there you can use.
which
Yeah,
process we can use at earlier stage?
like plant pathogenicity is there. You can use determine the pathogenicity or toxicity by using small fishes or plants. Okay, so all these things you should do.
Okay. Okay. Thank you, ma'am.
Okay, well,
Because it is, I think it is a major challenge for commercializing of microbial pigments, because even though it is a several advantages, only few microbial pigments are commercially available in market, like monoscous pigment and some carotenoid pigments only.
yes,
So
yes, yes,
toxicity is the, I think, is another aspect we have to consider when we work on the microbial pigments, because Pseudomonas is well known for the production of various kind of microbial pigments, but at the same time, the pycinin and other pyoidin like pigments are known for their toxicity. So I think the toxicity is the major challenge at the
yes,
same time.
yes. Actually, I'll say, sir, that to determine the toxicity of a given pigment, it is a first step in microbial pigment production research, sir. Once you confirm that given pigment it is non-toxic, then only you can proceed and you can go ahead for commercialization, sir.
Yes. Thank you, ma'am.
Yes, I also agree with Dr. Sunil because whether you're using it for textile or for as a food pigment, one has to see the toxicity. Otherwise, it will be difficult to go for commercialization.
Yes,
So,
yes,
so
ma'am.
that is there. Yes.
Actually, I
Yes,
think
ma'am.
I
Good evening.
forgot to write in the presentation about the toxicity, but it is a major challenge and it is an important factor actually.
Good evening, ma'am. Your presentation was very nice. This is Dr. Maya from Bangalore. So my concern was also the same. I was doing particular project in fungal pigments. So as Dr. Sunil sir and Madam from Delhi University has discussed now, first after the isolation, I have done the aflatoxin analysis, ma'am. So I think for fungal pigments, it will be well and good if you are conducting the mycotoxins that is present, so that only further we can just develop that work for food industry or any other industry. In fact, in
Right,
particularly when we are developing the color from the fungus. That
right.
is what I think. And madam, your presentation was very knowledgeable. Some of the techniques like NMR, all those things will definitely help in further research. Thank you so much, ma'am.
Thank you, ma'am. Thank you, ma'am. Yes,
There,
anyone else?
there is one question in the chat box by Krishna, if you can.
Okay, let me see. Just a minute.
I think it's can temperature and pH affect the microbial pigments?
Yes, yes. Who is Krishna? Yes, Krishna. Temperature and pH, it affects the microbial pigment production. I have already said that these two factors, they are very important. Okay.
Hello.
Yes, yes.
Yes, ma'am. I have one question. From which organism you have extracted your pigment? You just said I'm working since from last 5 years on pigment. So from which organism you have extracted pigment and in which media means whether it was a water soluble or soluble in methanol, ethanol, and what were your conclusion about that?
Okay, so I have worked on Pseudomonas aeruginosa, actually pyocyanin. Okay, it was extracellular. Okay, we produced it in a plain nutrient broth. Okay, and my conclusion is that actually we worked on the antimicrobial and antioxidant property of this pyocyanin, and then we found that cyanin, this pigment, it is I can say it is a potent antimicrobial and antioxidant agent. Okay, so this is my conclusion.
What about carcinogenicity testing? Have you tested cytotoxicity of that pigment?
Yes, yes, ma'am. We have tested. Uh, we did all these analysis, carcinogenic analysis. Okay, and we found that one fraction of our pigment, pyocyanin pigment, it was very effective in reducing the cancerous cells, ma'am.
One more point I want to discuss, ma'am. Whatever the amount of pigment produced by plants and my animals, these are in too much large quantity. But the microorganisms, as their production depends on the environmental condition, pH, temperature, and number of things, their production is not in that large quantity. So there are number of problems in production of microbial pigments. So
Yeah,
what?
yeah, ma'am. That is why I have stated this thing in major challenges. Actually, this is a major challenge. Many things they affect the microbial pigment production and yield. I've already told you all that yield of microbial microbes. Okay, it is also a major challenge. Okay, so we should work on these things. Like if you are having a pigment producing strain, we should work on things which can improve increase the yield, like mutation is there or many genetic level things are there, so we should work on those things.
And have you checked the stability of your pigment?
Yes, ma'am, we have done all these things which you are telling me, like stability studies and biological properties, chemical properties, structural determination properties, all these things we have done, ma'am.
Okay, okay.
Thank you.
Please share your address in chat box, please. Okay,
Yes, ma'am.
thank you.
Okay, ma'am. Okay.
Ma'am,
Okay.
how about UV protection? Did you work on that as well?
No, ma'am. Actually, we want to work on this thing. Really, will
Okay.
near future we'll work on this thing.
Sure, ma'am. Thank you.
Yes.
Good evening, Dr. Najia. I am Aparna Kumari from VIT Vellore. I am doing my PhD. For MSc student, I did some project with them. Then there also I isolate from actinomycetes, that is from hot spring sample. Just I want to know that those pigments can be responsible or can be connect with biomedical science like anti-cancer or some other thing. I just want to know, since you work for PhD, that's why you may guide
Yes,
us.
ma'am. Yes, ma'am. This is very, I'll say this is very good idea. Actually, many pigments, they have been reported to have many biological properties. Okay. Many researchers, they have reported that their pigment is showing anti-cancerous activity also. So I'll suggest that, yes, you can go with this thing, that you can connect your microbial pigments with the medical field.
Okay, ma'am. And one more thing, I just want to know that which color pigment you collected for your work, for your PhD work. I mean,
Yes,
which color?
ma'am. It was pyocyanin. Okay. So it was blue-green pigment.
Okay, okay. Okay, thank you so much, ma'am. And your presentation is really very helpful for whoever wants to work on microbial pigment in future.
Okay,
Thank
ma'am.
you so much, ma'am.
Okay, thanks a lot, ma'am.
Dr. Nazia, there are a few more questions in the chat box.
Do fungal pigments also suffer from stability issues? Yes, fungal pigments, they also suffer from stability issues. All microbial pigments, they suffer from the stability issues. Actually, bacterial and fungal pigments, majorly they suffer from the stability issues. Is there any particular microbe which can always give toxic pigments? Yes. Who is this? Actually, Kanare Yogini. No, no, no. This is Darji Parag. Okay. So many microbes, they have been reported to produce a particular kind of toxin. Okay. Uh, for example, pyocyanin. It is itself a toxic substance. So I'll say yes. Yes. Anyone else there?
Ma'am, I'm Manushree Haldar from GSS University, and I wanted to ask how to extract in.
Yes. Yes, ma'am.
I think she wants to ask how to extract intracellular pigment. This question is from Anushri Haldar.
Okay. So if your pigment is intracellular, 100% you need to get that pigment out from the microbial cells. Okay. You can use various methods, physical or mechanical methods. Okay. For breaking down the cells, like sonication is there. Okay. Mechanical breaking is there. So first thing is that you should break down those microbial cells, and then you can extract that microbial pigment by using the solvent, specifically non-polar solvents you can use. Yes.
I think,
Yes, ma'am. Thank you.
Thanks.
yes, now
Thanks.
we'll conclude.
Yes,
And
sir. Yes, sir.
I will
Yes.
request Aarti
Yes, sir.
to conclude it. But before that, again, I want to tell
Ha.
that this lecture is recorded and it will be circulated on the YouTube of Microbiology Society.
Thank you, sir. Thank you.
And second thing, if anyone is interested further to contact Nazia, you can ask me her email ID. I will provide to you, and you can increase your interaction. Thank
Yes,
you.
yes, sir.
Arti,
Definitely.
Yes, sir. Thank you, Dr. Nazia. I
Thank you.
wanted to express my gratitude for your insightful guest lecture on microbial pigment. Your expertise is on this subject and explanation. Once again, I will thank you for your sharing your knowledge with us. And we truly value for your contribution to your learning experience. I am also thankful Dr. A.M. Deshmukh who had given me opportunity. Thank you once again. Thank you MBSI. And here I declared that the session is over. Thank you once again.
Thank you.
again, one more declaration I want to make here.
Ha.
We are giving memento and certificate to all presenters. So I request among the listener, if anyone want to
Yes.
deliver a lecture on their PhD topic, only PhD topic. So that is our scheme. And I'm again requesting everyone, you can participate in that scheme. Thank you.
Thank you, sir. Thank you.